Imagine, imagine dialing 911 because your house is on fire, right? But the very moment you hit send, this invisible gas suddenly fills your room.
Just completely out of nowhere.
Exactly, out of nowhere.
Right.
And it doesn't break your phone, it doesn't cut the physical wires or anything like that. Instead, it literally alters the fundamental atmosphere of the room in this way that physically locks your phone's transmission signal inside the house.
Wow. Yeah, so the signal just never gets out.
Right. The dispatch center never hears a single thing, and, well, your house burns down.
It sounds totally like a bizarre sci-fi premise.
It really does. But a May 2026 paper in Science Advances just proved that this exact kind of molecular sabotage actually happens inside living organisms.
Which is just incredible.
It is. So welcome to today's deep dive. Our mission today is to explore this exact research, looking at how a highly reactive gas molecule can just, completely paralyze an entire organism's emergency response system.
It is honestly a phenomenal piece of biological detective work. I mean, the researchers were looking at this phenomenon in plants called systemic acquired resistance, or SAR.
Okay, right, sorry.
Yeah. And, well, we know that when a localized area of a plant gets infected by a pathogen, it generates this systemic signal. It travels through the vascular tissue, basically, to warn the rest of the uninfected plant to ramp up its defenses.
Like the plant's version of a broad-spectrum immune response.
Exactly. But the paper focuses on what happens when that systemic warning system inexplicably fails, and how a gas molecule, specifically nitric oxide, is the actual culprit behind the communication breakdown.
I always find it so fascinating how sophisticated plant immunity actually is. I mean, they don't have a circulatory system pumping specialized immune cells around like we do.
No white blood cells coming to the rescue.
Right, so everything relies entirely on chemical logistics. And the primary chemical distress signal we are tracking here, the 911 call, is salicylic acid, or SA. So in a normal, healthy scenario, an infection triggers a spike in salicylic acid at that local site, and then that SA gets loaded into the vascular highway to travel to the rest of the plant.
But the problem really arises when you introduce our second major player, right? Nitric oxide, or NO.
The invisible gas.
The invisible gas. Now, nitric oxide is normally an essential signaling molecule across almost all forms of life. I mean, it regulates growth, it handles stress responses. It's usually a good thing.
Okay.
But the researchers focused on a specific, highly mutated plant model. It's called the GSNOR1 mutant.
GSNOR1.
Yeah, GSNOR1. So in a standard organism, there's this constant, really delicate balancing act going on. An enzyme called GSNOR1 basically regulates the internal reservoir of nitric oxide by breaking down its storage form.
Oh, and the storage form is called S-nitrosoglutathione, right?
Exactly, S-nitrosoglutathione. It's a mouthful.
Okay, so let me guess. In this specific mutant line, that regulatory enzyme is just entirely missing or broken.
You nailed it. The enzyme is functionally defective. So because the plant can't break down that storage molecule, the GSNOR1 mutant accumulates massive, basically highly toxic levels of nitric oxide.
Wow. Okay, so it's just flooded with NO.
Flooded. And when researchers introduced a pathogen to these specific mutant plants, their systemic acquired resistance completely collapsed.
The 911 call gets blocked.
Right. The uninfected leaves never received the warning, and the whole plant became hypersusceptible to the infection. The localized defense was actually working fine, but that long-distance call just never made it.
Okay, let's unpack this for a second, because I'm hung up on this idea of an invisible gas physically trapping a chemical signal.
It's a weird concept.
It is. It sounds great in theory, but in a microscopic plant vein, how do you actually catch nitric oxide in the act of locking the door? I mean, is this like a fire alarm being so incredibly loud and chaotic that the firefighters can't actually hear the dispatcher's instructions? How do we know the signal isn't just getting, diluted or diverted somewhere else?
Well, what's fascinating here is that to map the exact location of the bottleneck, the researchers went straight to the plant's plumbing.
Okay.
They tapped into the pedial exudate, which is often called peak X.
Peak X.
Yeah. So if you isolate the stem of a leaf, the PIX is basically the vascular sap flowing out of it.
Right.
When they infected wild-type, totally healthy plants, they saw this massive influx of salicylic acid flooding into that sap, preparing to travel systemically.
Makes sense. The alarm is ringing.
But when they sampled the sap from the SNOR1 mutants under the exact same infection conditions, the vascular highway was practically empty.
Empty? Really?
Practically empty. The salicylic acid was being synthesized perfectly fine at the infection site, but it just wasn't making it into the transport vessels.
Okay, but let me push back a little.
Yeah.
Because correlation isn't causation. Right. just because the plant has high nitric oxide and an empty vascular highway doesn't necessarily mean the NO is the specific thing holding the SA back. I mean, there are a million cascading failures that could happen in a sick mutant plant.
Right, and that is exactly why they moved the investigation out of the whole plant entirely and went down to the single-cell level.
Oh, wow.
They designed this incredibly elegant experiment using protoplasts.
The ovoplasts.
Yeah.
Those are plant cells that have had their rigid outer cell walls stripped away, right?
Exactly.
Yeah.
Chemically stripped away, leaving basically just the plasma membrane. They wanted to isolate the exact mechanism of cellular uptake. But to test the variable, they needed to introduce nitric oxide artificially.
Wait, how do you artificially introduce a highly reactive transient gas into a delicate single-cell environment without just, well, destroying the cell entirely?
You use a chemical donor.
Okay.
So they applied a compound called detonanoate.
Etanoity.
Yeah, it reliably releases nitric oxide into the surrounding solution at a very controlled rate. And alongside that NO donor, they introduced a radioactively tagged version of salicylic acid.
Like as a tracker.
Exactly. Specifically, carbon-14 labeled SA. So this allowed them to measure precisely how much of the signal molecule was actually crossing the cell membrane.
And what happened?
The results were stark. As they increased the concentration of the NO donor, the cellular uptake of the radioactive SA was explicitly repressed.
Wow.
It was a perfect concentration-dependent suppression. Literally, the more nitric oxide in the environment, the less salicylic acid crossed the membrane.
Okay, wait. Let me push back on this for a second, too, because we established earlier that nitric oxide is a highly reactive free radical.
It is.
It notoriously loves to interact with and oxidize other molecules.
Mm.
So are we absolutely certain it's a transport issue? Isn't it highly probable that the NO is just chemically shredding the salicylic acid on contact before it even has a chance to cross the membrane?
See, the researchers anticipated that exact critique.
Oh, they did?
Yeah, because it is a very logical assumption that the NO is just destroying the messenger.
Right.
So to rule it out, they took the salicylic acid, incubated it heavily with the DETA-NOT donor, and then ran the resulting mixture through a mass spectrometer.
Looking at the structural integrity.
Exactly. They looked at the atomic structure of the SA post-exposure, and the mass spec data proved the SA molecule was totally intact.
Wait, really? Not damaged at all?
Not oxidized, not chemically altered in any way. The message was perfectly legible, the package was totally intact, but the loading dock doors were simply bolted shut.
That is wild. The package is sitting right there, completely undamaged, and the cell simply refuses to let it in.
Yep.
But if the cellular entry points at the leaf level are locked tight, how do the researchers figure out if the rest of the plant's immune system is also broken? I'd imagine a mutation that severe might just wreck the entire internal communication network, not just the front door.
And that became the next major hurdle. They basically had to play biological hackers and find a back door into the plant's vascular system.
A back door?
Yeah, they needed to bypass the local cellular membrane at the leaf surface entirely to see if the internal perception machinery, specifically the NPR1 protein pathways that actually read the SA signal, were still functional.
Okay, but I assume you can't just inject a microscopic plant vein with a tiny needle.
Right, no. So they initially tried a brute force approach. They took a massive concentration of SA, around 500 micromolar, and basically just sprayed it directly onto the leaves of the mutant plants.
Just douse them? Did it work?
It failed completely. The defense genes never activated, and when they tracked the SA, they found it was just pooling on the leaf surface. It was getting trapped in the extracellular space and the thick epicuticular waxes on the outside.
So spraying the leads is kind of like trying to deliver a pizza by just throwing it at a brick wall.
Exactly. The leaf surface transport mechanism was utterly impenetrable.
Here's where it gets really interesting then. So the front door is barricaded. Where's the back door?
They moved to a protocol called a soil drench.
Okay.
Instead of attacking the leaves, they flooded the root system with the salicylic acid solution.
Oh, because roots are different.
Yeah, the architecture of a root system is fundamentally different. It doesn't have the same transport barriers or that waxy cuticle layer that a leaf does, mostly because its primary job is bulk water and nutrient uptake.
So it's basically like walking the pizza through an open basement door directly to the elevator.
That is a perfect analogy. By soaking the roots, they successfully loaded the SA directly into the xylem, which is the plant's upward-flowing vascular tissue.
They bypassed the loading dock entirely and just put it straight onto the internal highway.
They did. And to confirm the SA was actually moving upward through the mutant plant, they measured the guttation droplets.
Wait, what are guttation droplets?
Those are the tiny droplets of xylem fluid that plants exude from the tips of their leaves. You can often see them in the early morning.
Oh yeah, like morning dew, but from inside the plant.
Exactly. And they found an 18-fold increase of salicylic acid in those leaf tip droplets.
An 18-fold increase reaching the very extremities of the plant.
Yes.
Which means the transport highway itself is perfectly clear. And if the SA is moving, does it actually trigger the alarm system?
It triggered it beautifully. That root-delivered SA successfully restored the mutant plant's systemic immunity. It literally flipped the switch on the PR1 defense genes, bringing the entire immune system back online.
Okay, so this proves something vital then. The mutant plant isn't fundamentally blind to the signal. The internal NPR1 receptors work perfectly.
Right. The catastrophic failure is isolated entirely to the specific membrane transport mechanisms at the leaf surface.
Man, this brings us to the core mystery of the whole thing. We know the nitric oxide isn't destroying the salicylic acid. We know the internal receptors still work. So how exactly is this invisible gas turning a perfectly permeable cellular membrane into a brick wall? I mean, I am struggling to understand the actual physics of how a gas molecule physically locks a membrane transporter.
Well, the mechanism comes down to one of the most powerful underlying drivers of biological transport.
Which is?
The pH gradient.
Ah, acidity.
Right. So to understand how nitric oxide was affecting the local environment, the researchers utilized these genetically encoded fluorescent sensors called fusion.
Oh, fusion with the capital H. I think I've read a bit about these sensors.
Oh, you have?
Yeah, they give like a ratiometric readout, right? So they aren't just glowing brighter or dimmer based on concentration. They actually shift their emission based on the surrounding proton density. So it gives you a live, real-time map of the acidity across the membrane.
Exactly. And that ratiometric reading is absolutely key to the precision of this experiment. These are incredible tools. They literally engineered the plants to express these sensors in very specific subcellular locations.
Okay.
Some were targeted to the apoplast, which is the extracellular space just outside the cell membrane, and others were targeted to the cytosol inside the cell.
Got it. So they're watching both sides of the door.
Right. And when they looked at the Greasenor-1 mutants, the sensors revealed a drastic disruption in the cellular environment. The excess nitric oxide was forcing the apoplast to become significantly more acidic, while simultaneously making the cytosol much more alkaline.
So it's literally a hostile environment.
Totally. The normal pH balance across the cellular membrane was completely thrown off.
Okay, so an acidic extracellular space and an alkaline interior. But why does a shift in protons suddenly paralyze a salicylic acid molecule? I mean, why can't it just diffuse across the membrane like other molecules do?
Because of its chemical nature. Salicylic acid is what we call a weak acid. It has a specific pK of about 2.97.
Okay, 2.97.
Yeah, and in the physiological pH of a plant cell, the vast majority of SA molecules exist in a charged state.
And charged molecules cannot simply slip through the lipid bilayer of a cell membrane.
Precisely. They require dedicated carrier proteins, specifically families of transporters like NPF, MATI, or ABCG. But those transporter proteins, they don't run on nothing. They require energy. And in this case, that energy comes from the proton motive force.
I think we need an analogy here because proton motive force can sound a bit, abstract.
Sure, yeah. Think of the whole system like a hydroelectric dam.
Okay, dam.
The cell membrane is the dam itself. The carrier proteins, those NPF and MATI transporters, they are the turbines built into the dam.
Right.
And the salicylic acid is the cargo that needs to be pushed through the turbines. But for a turbine to spin, it needs water pressure.
Right, the water has to flow.
Exactly. In a cell, that pressure is created by a high concentration of protons, so an acidic environment on one side, the membrane. The natural desire of those protons to basically rush across the membrane to equalize the pressure is what actually powers the transporter.
So by changing the acidity, the NO is essentially turning off the microscopic escalators that SA needs to cross the cell wall?
Exactly. It's a mechanism called proton cotransport. The SA basically hitches a ride on the physical rush of protons moving through the channel.
So if the nitric oxide is aggressively altering the pH on both sides of the membrane, it's essentially messing with the water levels at the dam.
It completely drains the reservoir. By driving the apoplast to become abnormally acidic and the cytosol to become alkaline in this, highly dysregulated way, the nitric oxide diminishes the specific proton motive force required for SA transport.
The water pressure drops.
The turbine stops spinning. And because those carrier proteins are powered down, the salicylic acid is physically trapped on the wrong side of the membrane.
That is just a staggering chain reaction. A gas accumulates, which disrupts the local proton gradient, which kills the power to the membrane transporters, which traps the distress signal, which leaves the entire organism defenseless against a microscopic pathogen.
It really is.
But wait, how do they prove definitively that it's the pH shift causing the traffic jam and not just some other random side effect of the toxic NO levels?
Oh, that's the best part. They validated it by testing a completely different mutant line known as the AHA1 mutant.
Oh, what?
Yeah. These plants do not have a mutation in their nitric oxide regulation. Their NO levels are totally normal.
Okay, so what's wrong with them?
They are missing a specific plasma membrane proton pump. So because that pump is missing, they cannot maintain their cellular pH gradients properly.
Oh.
Right. And when the researchers analyzed the AHA1 mutants, they found the exact same catastrophic failure. They had massively reduced SA in their vascular sap, and their systemic immunity was severely impaired.
That is brilliant. By breaking the proton pump directly in a totally different plant, they perfectly isolated the mechanism. It basically proves beyond a shadow of a doubt that if you break the pH gradient, you break the transport, regardless of how you did it.
It isolates the variable perfectly. If we connect this to the bigger picture, the nitric oxide is the trigger in the GSNOR1 plants, but the breakdown of the pH gradient is the actual weapon that locks the doors.
So what does this all mean? We've just spent a good chunk of time exploring the intricacies of a mutant mustard plant's vascular sap. And while the botanical detective work is genuinely thrilling, I think it's crucial to pull the lens back for a second.
Absolutely.
Why does this specific paper in Science Advances matter to someone listening to this deep dive who has absolutely zero interest in agriculture or plant biology? Why should you care?
It matters because biological mechanisms are highly conserved across kingdoms. I mean, we often view plants as these alien, distinct entities, but at a molecular level, they are solving the exact same physics and chemistry problems that human cells are solving.
Right.
Nitric oxide and pH dynamics are deeply intertwined in human physiology, too. In human endothelial cells, for example, acidic and alkaline conditions directly dictate the activity of human NO synthase, or NS.
Which does what?
That is the enzyme that controls vasodilation, the widening and narrowing of your blood vessels.
Are you kidding? So the exact same biochemical logic that controls sap flow in a leaf is actively controlling blood flow in my brain right now.
Entirely. And the connections go much deeper into pathology. When the human body fights an infection, our macrophage-mediated inflammatory response relies heavily on these exact same NO-pH interactions to traffic signaling molecules. And when that system becomes dysregulated, the consequences are devastating. In neurodegenerative conditions like Alzheimer's disease, aberrant nitric oxide levels actively impair the intracellular trafficking of neurotransmitters.
Seriously?
Yeah, it ruins synaptic activity using a very similar mechanism of transport disruption. We also see it in severe vascular disorders, where dysregulated NO limits the transport of vital vasoactive molecules across endothelial membranes.
It fundamentally changes how we can study these diseases then. I mean, we don't always need to rely on incredibly complex, highly restricted human tissue models to understand a cellular traffic jam.
That is exactly the greatest value of this research. Studying mutant plants like the JUXNOR1, or another line mentioned in the paper, the NOX1 mutant, which specifically overproduces NO in its chloroplasts, gives us these highly accessible, easily manipulable models for human pathophysiology. By studying how a plant cell fails to transport a weak acid under high NO conditions, we are directly dissecting the foundational mechanics of how NO-driven transport failures cause disease in humans. It basically provides a clean, observable window into cellular vulnerabilities.
It is the ultimate biological shortcut.
It really is.
So, looking at the big picture of this research to summarize for you, nitric oxide is an essential communicator. It's a molecule we literally cannot live without. But it operates on a razor's edge.
Very much so.
When that balance fails and NO accumulates, it wreaks absolute havoc on the local pH gradient. It diminishes the proton motive force, shuts down the membrane transporters, and essentially bolts the cellular doors shut. And in plants, this prevents the critical salicylic acid alarm from reaching the vascular highway, which leaves the organism totally vulnerable.
The balance is everything. The very molecule designed to act as an early warning system essentially becomes the agent that suffocates the secondary distress call.
As we wrap up today's deep dive, I want to leave you with a thought that builds on this research. Just something for you to explore on your own. It's a look at how we might eventually exploit this mechanism.
Oh, interesting.
We just spent this entire time talking about how excess NO, locking the cellular doors, is a catastrophic biological failure, right? And we discussed how researchers successfully hacked that system by bypassing the locked leaves and delivering the signal through the roots.
Right, the biological backdoor.
Exactly. But what if human medicine could harness that exact sabotage, but use it as a tool? If we know that highly localized bursts of nitric oxide can temporarily manipulate the pH gradient to shut down or alter membrane transport, could we artificially induce that state in specific human tissues?
Oh, wow. I see where you're going with this.
Imagine using a microscopic, targeted puff of NO to temporarily alter the pH around the blood-brain barrier. Could we use that transient shift to briefly open a cellular back door, allowing us to sneak notoriously difficult, life-saving Alzheimer's or cancer drugs right into the brain? Just like the researchers sneaked the salicylic acid through the roots of that mutant plant.
Basically weaponizing the traffic jam to force a completely different route.
Exactly.
That is a fascinating, deeply complex application of the mechanism. It really highlights just how powerful understanding these fundamental transport dynamics can be.
It is incredible what we can learn when we take the time to look closely at a broken system. So next time you're stuck in gridlock traffic or you hear an ambulance siren fading into the distance, just take a moment to appreciate the billions of microscopic alarms and cellular checkpoints operating flawlessly inside you right now. Thank you for joining us on this deep dive, and we'll see you on the next one.